Recording. Thank you, everyone, for coming. You know, the snow was starting to get me a little concerned about turnout. I can't thank you enough for just being good Canadians and being here in the snow. On that note, we are very, very grateful, and I will use the word privileged to have Dr. Jorge Ruas with us. No stranger to snow, a professor in Michigan at the University of Michigan in Ann Arbor, after several years of being a professor at Karolinska Institute in Stockholm, Sweden. And through there, he formed lots of connections that even overlap with our Muscle Health Research Center in kinesiology, like Dr. Arthur Cheng. They go way back. And prior to his position at Karolinska, he was a postdoctoral fellow at with Dr. Bruce Spiegelman in the USA, and that was seminal work on PTC1 alpha splice variants in muscle, and so on. And prior to that was graduate school also in Sweden. Is that right? Was that Karolinska as well?
That's Karolinska as well.
Karolinska.
Yeah.
So again, no stranger to snow. And you've come here to hear him talk, so I won't give you any clues about his presentation, but it's going to be very exciting. He gave a presentation yesterday at McMaster. Faculty at McMaster were texting me spoiler alerts about the talk, so I know things you don't know, and I'm very excited. But I think you'll be excited as well. So on that note, thank you so much for being here.
Thank you.
And it's all yours.
Let's see. Thank you very much. One day these presentations would be really plug and play, but they're not. Because as you all know, we just started using Zoom yesterday, so we're not used to it. So every single time on a lecture, on a class, everything, there's always computer doesn't recognize this and that. But I think we're good now. I never really practiced my talk. So when I said, I don't know what's coming next, we'll see what comes next. I put together a few slides about things that we have done in the past, because I thought that now that we're neighbors, it could stimulate ideas for collaboration. I am just also actively recruiting postdocs for the lab at the University of Michigan. I just moved in November 1st myself, just me. There is a lab back in Sweden that is just three and a half people, half a lab manager, a PhD student who is from Canada, a postdoc from Denmark, and a postdoc from Spain. And they will stay there another year and a half until everybody's done. And then I will close that lab and be at the University of Michigan exclusively. But I am recruiting for the University of Michigan lab at the moment. So I also thought if anyone is contemplating a postdoc, you get a flavor of what we do and how we think about these problems and the tools that we're using and so on. So the title is actually refers to the new study that I'm going to present, because I don't like to talk about only things that we have published. So I'm going to show you something that we have not submitted yet. It's in the process of submission. That is a story that I think took about 7 years or maybe longer to develop, because it was basically an observation from an RNA sequencing analysis that an amazing postdoc called Igor Chervenka made, and he slowly got to what I think is not only an interesting story, but the beginning of something more. And now let's see if it goes. I think if you click on it, it'll go. There we go. So yeah, very brief introduction. I'll have a few slides of things that are published. And as I said, it's just for you to have an idea of what we have been doing and how. I always say that this is kind of the goal of the lab. We are really looking to find molecular mediators of the benefits of exercise. And we look at secreted proteins, we look at secreted metabolites, we look at ligand receptor pairs, not only in skeletal muscle, but in different organs. We almost always start from a gene regulation place, because that is part of my PhD training and postdoc training. So we do a lot of what kind of pathways are changed in skeletal muscle, or in adipose tissue, or in liver, or in heart, or in brain, when you exercise, and how those pathways are often dysregulated in situations of disease. So we all appreciate that exercise is good for you, that you can use it as a prophylactic or preventive or therapeutic. The questions we are asking are why? And can we slowly trying to turn some of these discoveries in therapeutics that could help patients with insulin resistance or muscular dystrophy or a variety of other diseases? So I am not a believer in exercise in a pill, as a disclosure, but I am a believer that some of the discoveries we make along the way could be turned into therapies. And throughout the years, I have tried this a lot. I have a background in PharmD. We were just discussing during the interaction we had. So we are not a pharmacology lab per se, but I always try to see, is there a therapeutic future for our discoveries? Now, as you might know, and Chris mentioned, we started out as a skeletal muscle lab, and we started out focusing on PGC1 alpha coactivators, and we are still, as David was asking me, are you still going to work on PGC variants? Yes, I think so. This is a slide that Igor made for a review that we published already a few years ago, but I really like the busy slide because it summarizes on the one side many of the adaptations that we know occur in skeletal muscle with exercise, that being endurance exercise or resistance exercise, that have to do with angiogenesis, a more efficient delivery of oxygen and fuels, mitochondrial biogenesis and efficiency, ROS detoxification, a new and kind of really expanding and exploding field of inter-organ crosstalk, where people appreciate that a lot of the events that happen within the muscle fiber then are communicated both locally to other cell types within muscle, fibrodipogenic precursors, satellite cells, immune cells, but also secreted out to circulation to go talk to your fat, your liver, your heart, in the form of what we usually call myokines, or metabolite secretion. Of course, resistance exercise has specifically this kind of hypertrophy effect on muscle, and also this kind of improvement or the consolidation of the neuromuscular communication between motor neurons and muscle fibers, which I will give you some examples. Now, it doesn't mean that all of this is under the exclusive control of PGC1 coactivators, but over the years, a lot of the work that we have from in vitro systems, mouse genetic models, and also validation in human biopsies really show that as you exercise, your PGC coactivators go up. They enhance the transcription of different genes that indeed are involved in angiogenesis, mitochondrial biogenesis, and so on and so on. So we really like PGCs, and this is something that we put together with Bruce's lab a couple of years ago. This is one of those cell press snapshots that is like a one-page cheat sheet so that you can get as much information as you can about your favorite subject. They are for free. I think they're for free. And it's basically a resource for you to get into the field. This is an amazing work that Paulo Janig in my lab and Philip Domezic did in Bruce's lab, putting together everything we knew at the time about PGCs, from the gene regulation, what kind of transcription factors regulate the PGC1 alpha expression itself, what kind of isoforms or splicing variants we now appreciate that exist, and that's up to 20, I think. What kind of biology is linked to PGC1 alpha, which transcription factors are coactivated by PGC1 alpha and where do they bind, and post-translational modifications, kinases, phosphatases. So it's amazing that like one single gene and one class of coactivators has so much regulation from transcriptional, post-transcriptional, and post-translational modification. So I put it out there. If you are either interested or becoming interested in PGC1, I think this is a good resource to have. Now, we continue to be interested in PGC1. As I said, there's only one PGC1 alpha gene. Every species that has PGC1 alpha, and that goes on to the fly, there's only one gene, but this gene is transcribed in a very polymorphic way with different promoters, alternative splicing. So in the end, you get all these different variants that are transcripts, and some of them also different proteins linked at different biology. So we are still trying to understand, why do we need 20 PGCs? Do they all do the same? Is it about the regulation of the biological activity? But years ago, we also started using the tools that we have developed over the years, like transgenic mice for PGC that are born with high PGC amounts all the time, knockout mice for PGC that do not have the ability to express this gene, and asking questions like, if you modify skeletal muscle by overexpressing PGC or knocking it out, what happens to the rest of the body? And this kind of summarizes quite many years of studies that we found this new pathway that for us, it was new, and you can't really see the tight. I think there's a way to get this out of the way, right? Some kind of minimize or hide. There we go. So now the online people said, you disappeared. No.
Yeah, hopefully.
So as you can see here in the title, trained muscle kineresis and metabolism and mental health. So by using the PGC1 alpha transgenic mouse, which is a mouse that artificially expresses PGC1 alpha at high levels, basically at exercise levels from birth, and has a very distinctive phenotype that is, without any exercise training, that mouse can run about 100% more on a treadmill test than a wild-type mouse. So it's really very efficient at ATP production and oxygen delivery. So a little marathon runner in a way. We have used this mouse model to ask, can we spot biology in different organs? And our first study was to look at brain effects on mental health. And the reason for that was that it's very well appreciated that physical exercise, in particular endurance exercise or aerobic exercise, is a powerful tool to either prevent or treat certain types of depression, not all types of depression, but in particular stress-induced depression, that if you can get patients to comply to an exercise protocol, they really benefit from that. And the question has been, what is it about exercise that conveys that therapeutic effect? And there's many theories, and our discovery is not by any means the only one. People have talked about just endorphin release, about social reward, the fact that you have to, if you're living with depression, that you have to leave your home, go to the gym, interact with other people. All of this would have a positive reward. But we were interested in a specific question. Is there a muscle to brain communication through a soluble? We thought we were going to find a protective factor. We found the opposite. And what we found was indeed that if you exercise, and this has now been validated in mice, in rats, and in humans by many labs throughout the world, if you exercise, the levels of PGC1 alpha go up in your muscle fiber. And as a coactivator, PGC1 enhances the transcription of genes that are usually not expressed in muscle. They're called CATs. And people know that I like CATs because I was just chatting with some of you. CATs stands for kynurenine aminotransferases. So what they do is they take this tryptophan metabolite called kynurenine, which is a recognized neurotoxic compound that accumulates in the periphery and in the central nervous system under situations of high cortisol, stress, unpredictable stress. So this is not about being busy and running around. It's about something happening to you that you perceive as an unpredictable stress, and you cannot cope with that. So it's a different kind of stress. And in those situations, it generates a pro-inflammatory environment in your body, a high cortisol that induces the conversion of tryptophan into kynurenine. Kynurenine can cross the blood-brain barrier. So in those situations, it accumulates in the brain and gets further degraded into metabolites that are excitotoxic or very potent ROS generators or change synaptic plasticity. So in the field of psychiatry, it's very well appreciated that patients that suffer from depression, bipolar disorder, schizophrenia show high levels of these metabolites in their brain. And very, very linked with, I think, the modern explanation for some of these diseases, which is the concept of neuroinflammation, more than disturbance in serotonin as it was thought initially, or maybe complementary to that. So what we found is that if you exercise, or if in the case of our mouse models, if you have high levels of PGC1 alpha in your muscle, you start expressing 4 of these enzymes, these kynurenine aminotransferases. They're called CAT1, CAT2, CAT3, CAT4, very creatively. And they are very efficient converters of kynurenine into an acid form called kynurenic acid. So they remove the accumulating kynurenine from circulation. They convert it into kynurenic acid, which no longer has that ability to cross the blood-brain barrier. So what you're doing is preventing the accumulation in your brain of something which is excitotoxic and neurotoxic. So this was already 2014, and out of this, I became quite interested in what happens to kyna. So kynurenic acid was the first, or one of the first metabolites in this pathway that was discovered. The name kynurenic acid comes from the canis and urine, so it was found in dog urine, and it's thought to be an end metabolite of the pathway. So the idea is that it gets excreted through the urine, and that's that. Now, what we have seen during that study and after is that if you go for a run, this applies to a mouse, a rat, and a human. Those are the species that we looked at. With endurance exercise, kynurenic acid goes up in circulation, independently of your stress, cortisol, or inflammation. Exercise induces secretion of kynurenic acid from skeletal muscle, and it goes up in circulation. We all, regardless of species, have about 50 nanomolar kynurenic acid in circulation at baseline. And if you look at the well-trained person versus an untrained person, if you look at Arthur's blood and my blood, Arthur is going to have exactly the same level as me as baseline. But what happens is when Arthur goes for a run, he's going to have like a 10 to 12 fold elevation in kynurenic acid. If I go for a run, many other things will happen, but my kynurenic acid is not going to go up because I'm not a runner. So it's the elevation in kynurenic acid with exercise that is proportional to your fitness level. Now, when you go up from 50 nanomolar to 500 nanomolar, you are able to activate the GPCR, or G protein coupled receptor, that I'm sure you've heard about. This is a class of about 600 membrane receptors that represent one of the most actively targeted class of receptors by drugs in clinical use. They are ligand regulated, so they are amenable to pharmacology. In this case, GPR, they are called GPR and then a number. So you just have to memorize what kind of number it is. GPR35 is the receptor for kynurenic acid. And what we found is that GPR35 is expressed both in adipocytes and in immune cells So when you have this elevation in canurenic acid with exercise, you get both an energy expenditure, induction of lipolysis, what we call beiging or browning of adipose tissue, which I'm sure everybody has heard about brown fat and beige fat. So an increased energy expenditure, increased lipolysis, which makes a lot of sense because you're running, you need fatty acids to be sent to muscle. But we also see an anti-inflammatory phenotype in the population of immune cells that resides within the adipose tissue. So we don't see a lot of changes in circulation. We see that the immune cells that are recruited to the tissue tend to have a more M2 anti-inflammatory macrophage phenotype. So in essence, I once said this is kind of part of the biochemical explanation of the expression a sound mind in the sound body, because it explains in part how you protect your brain with exercise and how you harvest the anti-inflammatory effect and energy expenditure of the exercise. The other thing that we kind of found, which I thought, I always say, and it's true, this is one of my favorite papers that came out of our lab, and I really thought that it was going to fly higher, or not that that journal puts shame on anyone's family, but I thought this was going to fly higher. But I think people didn't appreciate that. It's very nerdy biochemistry, you know. It's about realizing something that I think evolutionary is very exciting, which is kynurenine is seen as a bad guy. It's basically a neurotoxic compound. If you accumulate kynurenine, it's usually related with cancer, psychiatric disease. What we found is that the same enzymes, the same CATs that I mentioned to you that are transaminases that convert kynurenine to kynurenic acid, the exact same enzymes, just given a different name, are some of the components of the malate aspartate shuttle, which is a shuttle that allows glycolysis to be more efficient. So this is based on the fact that the inner membrane of the mitochondria is not permeable to NADH. So if you run glycolysis, you have two options to renew that NAD. It's either you convert pyruvate to lactate, and you get your NADH to NAD, and the glycolysis can continue. But if you don't want to do that, you can import the NADH into the mitochondria for further ATP production, and you can do it through shuttles. This is one of the more efficient shuttles that increases your ATP production from 1 molecule of glucose by 50%, which for me is really exciting. The reviewers were not as excited as I was. But what I also think is really important is that it really shows that if you are a trained individual, something that is bad for you actually helps your muscle generate more energy. If you are an untrained individual, something that is bad for you is just bad for you. So really highlighting that the physiology of a trained individual versus untrained is very different. And also highlighting the fact that we tend to divide things into this is bad, this is good. It really depends on the biochemistry of the tissue that is prepared to convert this into something else and use it as an ATP generator versus just toxicity. Oh, we're stuck again. Here we go. So this is the pathway, and most people are more familiar with this upper part of the pathway where you see tryptophan, which is an essential amino acid, so we cannot produce it. We have to take it from diet. You most likely remember from school this upper part, which is tryptophan is a precursor to serotonin and then to melatonin. So these are mood regulators and sleep regulators. And this is probably one of the most well-known effects of tryptophan. Of course, you also need it for protein synthesis. In a non-exercised state, you use about 1% of your tryptophan for protein synthesis and about 4 to 5 for serotonin melatonin production. So 95% of your tryptophan is broken down through what is known as the kynurenine pathway of tryptophan degradation, or if you're cool, the KP. So what it is actually a de novo synthesis pathway for NAD. So that's how your cells make NAD from de novo. You have all the salvage pathways that are also very famous, but this is how you go from tryptophan, kynurenine, 3-hydroxykynurenine, 3-hydroxyandronilic acid, quinolinic acid, and NAD+. Now, the only cell type in your body, or one of the few, I shouldn't say the only, that is equipped with all the enzymes necessary to go from here to here are hepatocytes. Immune cells can do part, astrocytes can do part, neurons can do part, muscle can do part. But so it becomes an almost obligatory inter-organ communication pathway in the sense that one cell type brings it up to here, and then this metabolite goes to another place and gets broken down. So what happens in disease is that you get stuck in certain places. So if you accumulate, for example, 3-hydroxykynurenine, and this enzyme called kynurenine monooxygenase, or KMO, is extremely sensitive to inflammatory stimuli. So a pro-inflammatory environment is going to increase a lot KMO. 3-hydroxykynurenine is a very potent ROS generator, so it's cytotoxic. Quinolinic acid is an NMD receptor agonist, so it's neurotoxic. So it depends on the levels of all of these different metabolites. Now, nothing evolves just to be bad, you know, except one or two humans that I've come across throughout my life, but in biology, in general, things have a physiological role as well, right? So these are very interesting regulators of the immune system. So in the field of oncology, this is the part that people pay attention, which is quite interesting. So tumors, solid tumors, are known to secrete very high levels of kynurenine and kynurenic acid, which then act as ligands for an intracellular transcription factor / receptor called AHR, or dioxin receptor, depends on how old you are, that can regulate T cell fate. So the idea is that this is one of the mechanisms by which tumor cells can evade immune cell surveillance because they secrete kynurenine and kynurenic acid, they activate AHR inside the T cell and push it to a Treg state instead of a cytotoxic. So they can make the T cells allow the tumor to progress. And that's why there were and have been some clinical studies trying to inhibit the pathway at the level of tryptophan deoxygenase or indole dioxygenase. So basically stopping the conversion from tryptophan to kynurenine. And those clinical trials mostly failed. So people were a bit disappointed on it. And then a really cool paper from Christiane Opitz in Germany came up and said, well, if you stop it here, there's another enzyme called IL-4I1 that basically can also produce kynurenic acid. So, you know, these things are always redundant and complicated. So if you stop it in one way, cells find another way. So maybe there is still hope for this in terms of cytostatic therapies. And of course, I mentioned to you, our particular interest in this GPR35, which is activated by kynurenic acid. Now, another thing that we have discovered in terms of using PGC as a tool that I think is really interesting, I think that all our projects are interesting, so I just repeat myself. I think this is really interesting. We were looking at the fact that from the beginning, this was work from Bruce Spiegelman's lab, Christoph Hanschen in Switzerland, that really show that when you increase the levels of PGC1 alpha artificially or with exercise within the muscle fiber, there's a remodeling of the neuromuscular junction. Now, the question has been, it's a synapse, right? There's a motor neuron and a muscle fiber, and there's a synapse. So if you change the muscle fiber, how does the motor neuron know that the fiber has changed? So we initially collaborated with the bioengineering lab at KI, led by my friend Ana Teixeira, and using these microfluidic systems to culture cells and cultures of motor neurons and myotubes, we were able to identify this GDNF member called neuroturin as a PGC1 alpha related regulated myokine that induces motor neuron recruitment in an in vitro artificial system. And then we followed up this in vivo and published another paper showing that as muscle secretes neuroturin, which is a really quite short protein, a myokine, it has a dual effect. It acts back on the muscle fiber through its receptor GFR alpha 2 and induces kind of this glucose tolerance, oxidative metabolism, changes in calcium handling, oxidative fiber phenotype, but it also acts on the motor neuron to make them match the oxidative nature of the fiber. So I'm sure you all know your muscle physiology well, and there's this concept of motor unit homogeneity that a slow fiber innervates a slow motor neuron innervates a slow fiber, and a fast motor neuron innervates a fast fiber. And back in the 70s, people have done these experiments where they would take a slow motor neuron from a slow fiber and connect it to a fast fiber, and then the fiber turns slow as well. So kind of showing that this kind of motor neuron dominance over the fiber. In our case, we can show the opposite. As you change the fiber, the motor neuron also changes to become matching to this oxidative red type 1 fiber. Now, why is this interesting from a therapeutic perspective? In diseases such as amyotrophic lateral sclerosis, ALS, which is a motor neuron disease, it's perceived as a motor neuron disease. Your fast motor neurons are the ones to go first, and the slow motor neurons are conserved. So we are now exploring together with AstraZeneca and Alexion Pharmaceuticals if you can push this as a therapeutic that could help survival of those slow motor neurons if you can make more of them. And with the beneficial effects, both on the motor neuron side and the oxidative metabolism, we think that this could have interest for muscle injury, muscle dystrophies, and so on. So I'm a believer in this idea that small molecule PGC1 alpha activators, since I cannot see the clock, Chris, you have to yell at me when it's time for me to shut up.
Oh, we are not there.
Good. So I don't have any which is great. I love talking. So for me, it's just, I'm just gonna go on and on and on. So this is one of the first projects I started when I started the lab back in 2011, was to try to, you know, put your money where your mouth is in a way, like, can we find artificial ways to activate PGC1 alpha? And could those eventually become, eventually, tools for muscle atrophy, diabetes, obesity, and this really provocative idea that if your muscles are indeed participating in the protection of your mental health, could you imagine a class of drugs that targets your muscle to detoxify neurotoxic compounds instead of targeting your brain with all the side effects that antidepressive drugs have by targeting, which is of course like a very, very far-fetched, but I haven't given up on it. So Bruce and Novartis had done back in the day, which is published in PNAS, a small molecule screening for things that would induce transcription of the PGC1 alpha gene, and they found a few, but not too many. We thought that one of the reasons for that is that the PGC1 alpha protein is extremely unstable. So it has a half-life of about 30 minutes, which for transcriptional coactivator is fairly short. So what we thought was, can we design a screening platform that will find molecules that stabilize PGC1 alpha? So this is basically the work of Amanda Peterson, who was my first postdoc back in the day. And she designed the GFP fusion with different linkers in between GFP and PGC1 alpha so that this fusion protein keeps exactly the same half-life as the endogenous protein. And we have a cell line that basically at unstimulated states degrades this completely as PGC1 alpha would be degraded. And then when you treat it with different compounds, you stabilize PGC1 alpha. And this paper is published. So we could find about 30 compounds that in cells could stabilize PGC1 alpha, activate target genes, and increase respiration. So we had a molecular and a functional validation. Unfortunately, none of them was good enough to go into a mouse for solubility and toxicity issues. So the project was published as a proof of concept, but now that I am at the University of Michigan, that has a very famous unit for drug development, I've started talking to them. Is it worth rescreening again? Should we modify the compounds that we have? Can we find things that actually can go into a mouse? And eventually, if it doesn't work, I'll shut up. I probably won't shut up, but I'll continue. Yes, so as much as we like PGC and we continue to be interested in PGC, and we have more things coming out in terms of how these things work and why we need so many isoforms, we have been trying to rediscover other things that are not necessarily PGC dependent. Somehow in our projects, we always eventually fall on PGC, but I think it's just because we look for it and then it's there at some point. But we started this really interest in the lab about muscle mass regulation. So how does muscle go from eutrophy to atrophy to hypertrophy? What kind of genes, molecules are involved in that? And there's many systems to look at that. We use something that I learned from Leslie Lineone's lab a long time ago when I was a postdoc and visited her lab. She had, I don't think it was Leslie that invented this, but she had in the lab this hind limb suspension reloading or unloading reloading system that you might be familiar. It's a system where you modify your mouse cages or rat cages a little bit with some wires on top crossed. And then initially it was, you would put a ring through the tail of the mouse and that suspend that with a nylon thread. We have since modified it for no surgery at all. So just with surgical tape, you can fix the nylon thread to the base of the mouse and that it's fine. It stays there and you avoid the ring. So what you do is you set it up in a way that the mice are slightly elevated from the ground, so the hind limbs don't touch the ground. The angle is not as severe. So they have radial mobility. They can access their food, their water, but the hind limbs don't touch the ground. So that leads to a 30% muscle mass loss within a period of 10 days. So you can study in a very time-controlled way the process of atrophy. At the end of that, you can also disassemble the setup and the mouse has to regain mobility. So the reloading causes a compensatory. It's a mixture of tissue regeneration and hypertrophy. So it's not the same pure hypertrophy that you get if you would pump iron. But we study that as a compensatory hypertrophy, and you need another 10 days to get more or less to the same level. And it is one of the few systems that allows you to study both the atrophy and the hypertrophy versus control in the same setup. You could also do synergistic ablation of muscle. Some people do that, but and what we do that is then we obtain samples that are from other species like rats, or we obtain human biopsies, muscle biopsies from people that have been on long-term bed rest, intensive care unit. They may have atrophy as muscle atrophy as a comorbidity of a heart disease. So we kind of cross-validate things between these two settings. So what we have done is RNA sequencing, gene expression analysis on this control, atrophy, or the first day of reloading. So we have now done first 1 day, 3 days, and 7 days, but the beginning of this study was just 1 day where you do not see any hypertrophy. So our idea was, can we catch the beginning of things? What are the first pathways to be activated that then allow muscle to regenerate and go in and regain muscle mass and regain muscle function? And a lot of projects have come out of this. Almost all of them took a while. Somebody was just asking me about what would be the expected duration of a postdoc, you know, and if you want to take something like this study that was led by Jorge, who was in the lab at the time, he really became interested on this gene that is annotated on the genome as zinc finger protein 697. There's no literature on it. There's no information on it. If you go on PubMed, you will find a GWAS paper that connects the locus of ZFP 697 to multiple sclerosis, but that's it. There's no other papers. And Jorge was also the first author on the neutrin paper. So he came to me and said, this gene goes up a lot in the first day of reloading. Can I try to understand what it does? And this is a very risky project. So if it was his only project, I would probably not have supported the idea, but it was not. So he spent a lot of time figuring out what this does. And the challenges are there are no antibodies, there's no mouse model, there's no reagents, you have to make everything yourself. So this took 6-7 years to get to fruition. We are now revising it for publication. It has taken a little bit less traction. I always think that our papers are amazing and that the journals with small names are going to really... Lately, I have been less in line with what they want, but I hope it will end up in a good place with visibility because I think it really deserves. It's on bioRxiv, so I'm not going to say anything else about it, except we found it to be an RNA binding protein that is involved in how muscle fibers secrete cytokines to talk to fibroadipogenic precursors and satellite cells and immune cells, which is a conversation that must happen if you want to have muscle regeneration and hypertrophy. And we saw that in our ZFP 697 muscle knockout, so ZFP is only knocked out in the muscle fiber. If you stop the conversation there, muscle cannot regenerate. So for many things, I think this is interesting, including the fact that we know that satellite cells are important for regeneration, immune cells are important, FABs are important. I think it's quite interesting that if you prevent the muscle fiber from calling for help in a way, by not secreting those cytokines, the conversation stops and you don't have regeneration. Now, we are not going to rename it. We thought we would rename it. And the guys in the lab came up with this acronym because ZFPs are all called ZFP and a number. So it's virtually impossible for you to remember which ZFP does what. So we thought we would call it TRAIN for tissue remodeler and activator of inflammation. But over the years, I've realized that sometimes if you rename something, people get all agitated and nervous. So ZFP 697, it is. Now, a study that just came out, and we were chatting about it briefly, also came out of this screening, and this was another postdoc in the lab's favorite gene. The postdoc is called Serge Ducomont from Switzerland, and Serge became interested in this Mustang protein that was not cloned by us. This was cloned by the lab of Michael, and I will butcher his last name, Argiru, I think, with my apologies for the mispronunciation of the name. And so Michael had shown that Mustang, as he called it, is a protein involved in muscle differentiation and myoblast differentiation. We saw it really, really upregulated in the reloading stage. So this Mustang over here, and Serge really, I mean, he did an amazing job trying to figure out what this thing was doing. This is a micropeptide, so it's 82 amino acids long, extremely well conserved between species, that we actually found to be highly expressed, not only in skeletal muscle, but in smooth muscle. So if you see here, it's a mouse tissue and human tissue panel, you see that indeed skeletal muscle expresses high levels, but smooth muscle-rich tissues like aorta, tibial artery, they have really the highest levels of Mustang. And after trying everything under the moon, we made 2 whole body knockouts. The first was leaky, we couldn't use it, we had to make another one. Then we made a skeletal muscle knockout, and then we figured out that it was not coming from the muscle fiber, it was coming from smooth muscle. And indeed, you can see here on the staining that this is a wild type mouse, this is a Mustang knockout, and you can see this, the signal of Mustang comes mainly from the bigger blood vessels in the muscle, not from all of them. This is an alpha smooth muscle acting as a counter staining. So what Serge actually discovered is that this is actually secreted from the smooth muscle cell, and it talks to the muscle extracellular matrix to really modify the ECM, which is a necessary step for the muscle remodeling, for the fibers to go bigger. We couldn't, for all kinds of reasons, take it any further, so we don't really know how it works. But if you knock out Mustang, we see a sexual dimorphism in terms of developing fibrosis after injury. So females develop more fibrosis than males if you induce muscle injury. Okay, now for the new things. As I mentioned, this is a picture of Igor, always very serious. Arthur knows Igor as well. So Igor really changed the way that we did our RNA sequencing analysis in the lab. He really likes informatics and bioinformatics. So he really set himself from the beginning of the postdoc to develop scripts and R's and you know, R scripts and pipelines for us to do our own analysis, which really changed how we do things. So he had another idea. Instead of using the PGC mice or the unloading, reloading, he said, how about I find RNA sequencing data sets that I can download as the raw data and reanalyze everything myself. So he went for human myopathies and mouse models of atrophy. His idea is, can I find something that is so fundamentally conserved that is important for humans, for mice, for myopathy and atrophy? And so this is as much as he could find. So you would expect maybe he could find more data sets, but he was really going for things that he had access to the raw data, so that he could download and reanalyze everything with the same parameters, because everybody analyzes their RNA sequencing slightly different. And he wanted things that were comparable, done in the same way. So in the end, he has this 6 of myopathies and 7 or 6 as well of muscle atrophy. And he used these upset plots that basically look in what's in common between set 1 and 2 and 1 and 3 and 1 and 4. And this, of course, generates an enormous amount of information. And it is now a really valuable resource for the lab, because somebody can come in and say, oh, I really want to know what's interesting between critical limb ischemia and myotonic dystrophy and so on. But his question was, first, can we actually see in these data sets things that we know should be there? In other words, does the approach work? And although this is very small font, what it is genes that we know should be up in situations of atrophy, genes that we know should be down in terms of mitochondrial dysfunction, up in regeneration and up in fibrosis. So these are just, can you find your favorite gene in there? This was just a validation, not like the sanity check. And then he asked, what's in common? And if you ask what is in common between all of these sets, there's only one gene that is in common, and it's called HECD1. And that's the story I'm going to tell you in the remaining of the time. So what is HECD1? HECD1 is an E3 ubiquitin ligase. And sure, you know your ubiquitin proteasome degradation pathways. These are E3 ligases are very famous in muscle atrophy. They are usually connected to protein degradation through a system that involves several steps. Like there's an E1, which activates ubiquitin. Ubiquitin is a small molecule that is going to be eventually be added to your substrate. And when substrates are polyubiquitinated, they get degraded by the proteasome. And the proteasome chops one amino acid at a time, and you can recycle everything again. So this pathway is mostly famous in the field of protein degradation, but not only. So you have about a couple of E1, so they're very general, they activate ubiquitin. You have about tens of E2s. The E2s transfer ubiquitin to the E3, and you have about hundreds of E3s, and E3s are the ones that give substrate specificity. So these E3s are only going to recognize specific substrates, and then they transfer ubiquitin to the substrate, polyubiquitination. This leads to degradation. And this is, of course, how during muscle atrophy, the sarcomere, for example, is degraded and disassembled. And there's many other examples of protein control by protein ubiquitination. Now, although this is definitely the most famous function for protein ubiquitination, different ubiquitination of the substrates can be connected to autophagy, cell signaling, endocytosis, DNA damage. So there's a variety of cellular processes that are controlled by substrate ubiquitination that are not only protein degradation. They have mostly to do with the fact that these side chains of ubiquitin, where the first ubiquitin is added to a lysine, and then ubiquitins can be added to other ubiquitins, these chains can be built in different ways. So I'm not going to go through all of them, but you can build them on lysine 6, on lysine 11, on 27, or you can have branched, you can have mixed. So depending on the build of these side chains, you can then have different biological activities. Now, we found HECD1 to be dysregulated in all of the data sets that we studied, both myopathy and atrophy, but it turns out that it's dysregulated in a differential way. So we see it to be increased in gene expression in models of atrophy and decreased in gene expression in models of myopathy. It belongs to a large family of E3 ligases. They are all part of this family because they have a HECD domain. HECD1 is a very large protein of about 300 kilodaltons, so it's a quite significant challenge to work it in terms of molecular biology, overexpression, and so on. And AlphaFold, that I'm sure you are familiar with, says that it looks like this. I don't know if it does because this is a prediction, but it's all of these different ubiquitin ligases from the HECT family have different functions. They have different domain composition, and you can find HECD1 over here. Now, there's not a lot known about HECD1 because the knockout is embryonic lethal. So if you knock out HECD1 in the whole body, the neural tube doesn't close, mostly because you interfere with cell migration for that closure. So those mice are not viable. There's some GWAS indications about cognition, about neurological disease, but nothing super, super strong. It is not exclusively expressed in skeletal muscle. So here you have in orange different kinds of muscles, but then you can see also liver, kidney, heart, and so on. This is mouse, this is human, and this is a Western blot to complement this gene expression pattern that you can see over here. And there are one or two things that I want to call your attention. So the first one is that the band you're looking for is the full-length HECD1, which is about 290 kilodalton. So it's the one over here. And this is going to become important in a second when I show you the knockout data. But we also see all of these other bands. Now, this could very well be because the antibody is not super specific and recognizes other things, but this is a gene that has 46 exons, I think. So it's a very large gene. So the possibility of alternative splicing is also high. So these could be splice forms. They could be something else. We never looked into this, but I'll call your attention again to this in a second. How is it regulated? So in addition to the gene expression in myopathies and atrophy, we looked at data sets that we had in the lab. This is our unloading reloading system. And again, it goes up in atrophy, down in the regeneration stage, down in regeneration induced by muscle injury. This is the only data set. We have many more. We looked at human interventions, mouse interventions, all kinds of things. And the image that always comes out is it goes up in situations of muscle atrophy and down in situations of muscle regeneration, except downhill running, which we perceive as something that induces muscle injury. And we would expect it to also go down, but it goes up for whatever reason. Now, the first gain of function, loss of function thing that we did with HECD1 was to do a knockdown in C2, C12 myotubes. So I always make it very clear that I'm not a fan of C2, C12 myotubes, but we did it for a reason. There was a technical that we couldn't get it to work properly in primary myotubes. There's an additional phenotype when you do it in primary myotubes that we haven't explored yet. And that is, if you knock down HECD1 in primary myoblasts, they all fuse together and they make a gigantic myotube, which it's not the myomaker. We looked at that, which we don't know what it means. And we will definitely try to figure that out. But it was not possible to do this experiment at that system. So we used differentiated C12 myotubes with a knockdown of HECD1. Now, being an E3 ligase, we went for a proteomics approach because you would expect that it would control protein levels. And so we got a lot of help from Marta Murgia, who is she has a lab in Italy, and she's also in Matthias Mann's lab in Germany. So Marta did all of the proteomics for us. And the first surprise is this. So I just said an E3 ligase in muscle is supposed or known to degrade proteins. Now, when we knock down HECD1, when we reduce the levels of HECD1, we see less abundance of all kinds of proteins related to muscle contraction, muscle differentiation, myofibril assembly, which is not exactly what you would expect, right? If it's degrading protein and it goes down, your protein should go up. So we just see a down regulation of all kinds of cytoskeletal sarcomere assembly proteins, which we thought was kind of interesting. Now, I am sure you have seen this. I bring it up for two reasons, because I just think that the electron microscopy of a sarcomere is quite pretty. And it has this really nicely organized Z line and the H zone, all of this banding, but also for you to appreciate in a second what happens when you knock out HECD1 I'm so fixed on PGC1. What happens when you knock out HECD1 in skeletal muscle? And the other reason was that we looked at all the different components, from the thin elements and the thick elements. We have here all the different components. Now, Igor decided to use a String, the software that you can use to do pathway analysis. I don't know if you've ever used it, but it's basically an online free software that will take a list of genes that it's designed for gene expression. So that's why the trick here. But you can upload your list of genes, and then String is annotated on pathways. So it's going to group your genes or proteins in pathways that have been published to be related, interacting, functionally annotated. So it's important to look at that. You'll see in a second that it gives you little clusters of circles that are connected by lines. The color of that line has a code, so pay attention to that, because it could be experimentally validated, literature mining, function. So there's a it doesn't mean that everybody has validated the interaction. But it is one way for you to group your genes of interest. Now, as I said, string is developed for gene expression annotation. So the challenge you have when you do proteomics is that unfortunately, not all protein names are the same as the gene names. I don't know if you ever, there's a couple of not. So what you have to do first is convert all your proteomics list to the corresponding gene names or vice versa, and then annotate it to the system. What it happens is you get something like this. There's different color coding here. So the first thing I'll call your attention is the out circle on every single protein. If it's blue, it means that that protein is less represented, so down. If it's red, it means that that protein is more represented in the proteomics data set. And then the cluster annotation, which is the inside color, that is done by the software, meaning that these proteins are related, and these proteins are related, and these proteins are related, and so on. HECD1 is over here. So it has a blue circle around it. That makes sense. It was a knockdown, so it's reduced. Now, the really first kind of image you get out of this is this enormous cluster of proteins. Now, if you remember the acronyms for the sarcomere proteins, TPN, TNNI, TNNC, TNNT, you look here, they're all here. Not all of them, but almost all. So there's titin, there's myosin heavy chains, there's tropomyosin. So what this means is that in the absence of HECD1 expression, sarcomeric proteins go down, which was kind of unexpected. The other kind of surprise here, which was a little bit more specialized and required some homework, was that the protein that links HECD1 to the sarcomere proteins cluster is a protein called KLHL41. So I don't know if anyone is interested in myopathies here, but these proteins called KLHL41 and KLHL40 are chaperones that are involved in Nemelin myopathy. So this is a form of myopathy that basically is caused by either mutations on these chaperones or on their target called nebulin. So if you have mutations on KLHL or actinin or nebulin, what happens is that the thin element of the sarcomere cannot assemble, the sarcomere collapses. So you have, depending on the mutation, you can have like severe congenital and after birth manifestations, or you can have progressive diseases that sometimes show up when you're 40 or 50 years old. They start manifesting by distal weakness and eventually lead to loss of locomotion and paralysis and also respiratory problems. So this was kind of interesting. And the model about Nemelin myopathy that we appreciate today is the following. You have KLHL 40 or 41. These are two members of the same family. They need to be ubiquitinated by an unknown E3 ligase. So I guess you know where I'm going with this now. Once they are ubiquitinated, they get activated as a chaperone that promotes the assembly of nebulin from Z line to Z line almost. Whereas if you do not have activation of KLHL41 or in patients you have mutations, nebulin aggregates and the sarcomere collapses. Now, the reason for this strict dependency on sarcomere assembly in nebulin is that nebulin is a large protein of about 600 to 900 kilodalton, depending on the form. And you can see it here represented in pink. And it's tightly regulated and tightly bound to the Z line. So it's very structural. It binds to other very structural proteins on the Z line, but it also is required for the assembly of the entire thin element. So if you don't have nebulin polymerization, actin doesn't polymerize and none of the other proteins polymerize and it basically collapses. So this is what an electron microscopy picture from muscle from people with Nemelin myopathy will look. They don't always look the same. It depends if you have mutations on actin, on KLHL41, cofilin, lyomodulin. The classical histology diagnostic for Nemelin myopathy is what is called Nemelin rods, which is observed both in EM, but also in regular histology sections. And it's these dark condensates that are formed by the aggregation of nebulin. Now, not all forms of Nemelin myopathy show Nemelin rods. You can see them here, you can see them here, you can see them here, less here. But what you can appreciate, and now if you recall the beautiful image of the sarcomere, is that it is completely disorganized. You have lost the nice banding and organization here, dramatically so here, and in here and in here. And this is, of course, what leads to progressive muscle weakness and dysfunction. You could also look at the list of genes that are known to cause Nemelin myopathy. They are over here. There are about 13 genes where mutations have been found to be related to Nemelin myopathy. We find that about 6 of them are affected by the absence of HECD1. So in our proteomics data set. Of course, as I said, it's known that KLHL41 needs to be ubiquitinated to be activated. So what Igor did was this not easy experiments that require overexpressing every single protein in the system to try to figure out if indeed HECD1 could stabilize KLHL41. And you can see it here that a wild type HECD1 can, but a mutant from HECD1 cannot. It's quantified over here. He also showed that it stabilizes because it ubiquitinates. So this is a ubiquitination assay where you overexpress this HECD1, wild type and mutant, flag and ubiquitin, and you can see that the levels of ubiquitination increase. If you're not familiar with these Western blots, they look kind of ugly because what you're looking is that different lengths of chains for the same protein. So more ubiquitin and more, so you see this kind of ladder effect that you can see over here. He also has a very interesting observation that we cannot say for sure it's a direct effect of HECD1 on nebulin, but he tried to see if indeed nebulin got stabilized by overexpressing all the components. And you can see here that if you have no HECD1 versus having HECD1, nebulin gets quite stabilized in the presence of KLHL41, but you also see a nice stabilization in the absence of KLHL41. So now we cannot guarantee that it's not because these cells expressed a little bit of KLHL41 that was sufficient, but it could also be that HECD1 could have a direct effect on nebulin, which could also be explored further. Now I'm getting to the end. I'm going to show you just the phenotype of the mouse model that we have generated. As I mentioned, HECD1 is a very large gene. It's on the minus strand of the genome, so it's in this direction over here. And I forget the exact number of exons, but 40 plus exons. We acquired the first allele from the IMPC, the International Mouse Phenotyping Consortium, which, if you're not familiar with, it's a network of labs that are floxing every single gene in the genome. They are funded by different countries, by the European Union. So you can get that mouse model from them for about, I think, $5,000 that you can get the mouse model. They always make their mice in the same way. So they select a particular exon, and they do a good job looking at which exon they should flox. They put LOXP sites around it, but they do it in a knockout first strategy, which basically has this LAC-Z neomycin cassette. So what it means is that the mouse that you get from them is a knockout first model that forces the splicing of exon 2, in this case, instead of going exon 3, 4, 5, it goes into this cassette. So you have a knockout, but you also have a reporter because LAC-Z is going to be expressed wherever your gene would be. So that could be interesting. But if you want to do a tissue-specific knockout, you can cross this allele, this mouse, first with the flip recombinase that removes green to green. So the neomycin cassette like it disappears. You get your nice floxed allele, which you then can cross with a CRE specific to whatever tissue. In this case, we use a human alpha skeletal actin to get skeletal muscle specific HECD1 knockout. What this causes, the deletion of exon 3, it's a very premature stop codon in the very beginning of the protein. This is the catalytic domain over here. And that's why I showed you the Western blot, because if you now look at the knockout, you have your floxed control, this is your knockout. And what you see is that indeed the 290 kilodalton band is gone, but these guys are still here. So we don't know if it's a split. We thought it's very unlikely that these are big enough to constitute the HECD catalytic domain. And in addition, HECD1 needs not only the catalytic domain, but the docking domain. So we don't think that this would generate any kind of hypomorph, but just to put it out there and alert you for that. The mice are born in the right sex percentages, Mendelian ratios, females and males. So body mass is the same. Body composition is the same. They are weaker. So these are young mice at 3 months of age, 3-4 months of age. You can see already in grip strength, which indeed is a very general evaluation of grip strength, but you can already see that both males and females show signs of muscle weakness. They also show signs of glucose intolerance. So we can already see some pre-diabetes situation in this, but at this age, this reduction in muscle weakness and glucose intolerance is not sufficient to affect exercise performance. So on a run to exhaustion on the treadmill, they still perform exactly the same. So we see muscle weakness, some metabolic effects, but no exercise performance. We also, because of this muscle weakness, we started thinking, is there a selective loss of certain muscle fibers? In humans, nemaline myopathy can be a type 1 nemaline myopathy or a type 2. So people in humans can have a disease that is preferentially on their oxidative fibers or their glycolytic fibers. So we thought maybe there's a phenotype in there. In our mouse model, it seems to be a more generalized, like a confused fiber that is trying to regain a myosin heavy chain identity. And indeed, what we see is that there's a quite significant increase of embryonic myosin heavy chain and myogenin, which are signs of constant regeneration that has not been resolved yet. So this also means that this muscle is not weaker and insulin resistant. It's also remodeling itself in a way that is not resolved eventually. You can do RNA sequencing on this. You can see all kinds of genes related to enamel myopathy. But one surprise was that we see a significant downregulation of mitochondrial genes, which is not associated with this disease. So talking with clinicians throughout the world, some at KI, some at the Mayo Clinic, we really reached out to anyone that was working with these patients and asked, do you see mitochondrial dysfunction in these patients? And they said, it's not something we look at. But as I said, I mean, this, in a way, it could be because it's not there. It could be because these patients have other problems that immediately need to be more solved in the clinic, which is the muscle weakness. And so in our case, we tested this in transcriptomics. We tested this in, this is pathway analysis. You can again see downregulation of oxidative metabolism. We tried to see upstream if we could find, is there evidence of dysregulation of transcription factors that could be justifying these? And indeed, we find them. We saw, for example, for the downregulated genes, the predicted regulators are ERR, YY1, but this could also be an indirect compensatory effect. You can validate it in a more traditional way. This is electron transport chain Western blot, like I'm sure you've seen before, and you see there's a downregulation of all of this. And this is a genomic DNA, and you can also do it in a more stain. So any way that we try, mitochondrial dysfunction is there, is just not usually associated with enamel myopathy. Now, the proteomics of muscle catches both the mitochondrial dysfunction as the RNA sequencing catch. So we catch the mitochondrial dysfunction both at gene expression and protein expression. So kind of suggesting to us, this is not a direct effect of the HECD1 absence. This is probably an indirect effect, because otherwise you would see it at the protein level, but not at the gene expression level. And this is just comparing the two things. So what does the muscle actually look like? So this is a flox allele. I'm done. Flox allele, the wild type, and you have the nice organization. These are different images from a control mouse. You have your nice mitochondria here in between the muscle. If you look at the knockout, it's kind of full of holes. And this is really striking. I was really surprised. At first, I thought the facility messed up because this is not normal. We've never seen this. And also the other thing is that these mice are okay. I mean, I showed you this, they're a little bit weaker and glucose, but on an exercise treadmill, they're fine. And you see not only this disarrangement of the sarcomere, there's not, the banding is not there here, super disarranged. There's also a fiber type dependent. So we don't see this in every single fiber. You can have a fiber that is totally fine, and the next one shows like this. But many of the mitochondria are absent. Now, again, talking to the clinicians, they said this is not abnormal. They see that sometimes when they catch the patients at 40 years of age or 50 years of age, they've been okay. So when they look at the muscle, it's going to look as disarranged as this, and the person only perceives that there's a disease at that stage. So it's amazing that muscles can coordinate and survive with this degree of morphological disarrangement. Now, at 1 year of age, because of this age dependency of the disease, you can now see even a more profound reduction in muscle strength and already significant reduction in exercise performance. And at this stage, I had an idea to ask a histopathology lab at the Mayo Clinic that works with these kind of patients and ask, would you consider taking our mouse knockout samples and running the histology tests that you run for humans? And Eli Nadaf, who is a clinician at Mayo Clinic and responsible for this, he said, I'll do it. So he ran tens of tests, including the ones that we had read. He ran them again. So he sees again this mitochondrial dysfunction. This is the staining you would use to see the enamelin rods, and it does not see enamelin rods. So this form, our model is not showing enamelin rods. But what he found was something quite interesting, that he really had the, I know I should end, you're right. I'm almost there. He basically found this really interesting desmin accumulation. So for the heart people, this becomes more interesting. This really, that foci of desmin accumulation that he thought was so interesting that he sent for electron microscopy. So what you see here is that desmin, which is a very large filamentous protein that has the role of not only binding lateral sarcomeres, but binding nebulin to the Z line and then connecting to mitochondria and to the nucleus. So it's basically a network that keeps the sarcomeres together. It's broken down and it's accumulating in these rods in these areas. So in essence, this is kind of illustration of what desmin represented in red is kind of doing. Here you can see that it's both connecting laterally, but also to the mitochondria and the nucleus. So we kind of think that the mitochondrial effect that we see could very well be a consequence of the destruction of these desmin filaments, so that when the sarcomere is so disorganized, that mitochondria that are so tightly connected in skeletal muscle and in heart muscle are going to be disorganized as well. So this is the summary of what we see things like. This is, I always say very proudly, this is my own drawing from BioRender. So please do not criticize. And this is the best that I could do for a dysfunctional mitochondria. So the idea, the model that we have is that HECD1 is an E3 ligase that ubiquitinates both KLHL40-41, but instead of having a protein stability role or a protein degradation role, it actually works as a chaperone, activates them, which in turn are chaperones for nebulin that can polymerize and things are kind of in place. We don't know if HECD1 has a direct effect on nebulin or a direct effect on desmin, but what we see is that if HECD1 is not there, this doesn't get ubiquitinated and the whole thing aggregates and collapses together with mitochondrial dysfunction. So the last thing is, are there humans out there with HECD1 mutations? And as I said, you cannot have complete loss of function of HECD1 because it's embryonic lethal. So you will never find those. But we contacted the MyoSeq database in the UK, so Volker Staub and Anna Topf, who were very, very kind and looked as hard as they could. And they found a family that carries an alanine to valine mutation in amino acid 1006. And there's, unfortunately, if you read the last line, it says, unfortunately, the records related to these diagnostics were lost. The way that these networks work is they have the clinics from which they only retrieve the clinical data when it's interesting. And in this case, but it's interesting that the father had been, both the father and the son diagnosed with this Aaron muscular atrophy, which is kind of an exclusion diagnostic when you don't know the genetic cause. The father was a boxer, actually, so very active in exercise, and developed on his 30s, developed muscle weakness that eventually progressed. So we are now trying to find other data sets to see how prevalent are these mutations in HECD1. And this is it. I just want to highlight again, I think I mentioned our main collaborators as I was going through, but I really want to say that we don't do things by ourselves. We always depend on collaborations. And to show you a picture of what the lab looked like 2 Christmases ago, this is Igor over here. This is Serge that I mentioned for the Mustang paper, and Paulo that I mentioned for the Cell Press snapshot. This is Kyle, the Canadian PhD student that everybody calls Canadian Kyle, because it flows. The lab doesn't look like this anymore. And if you are interested in our projects, and you can find our website, which is designed and made by Igor as well. And it's done in a very popular science way. So our idea with the website was to really show people what do we do, who we are. And these are the people that are left back in Sweden. And as I said, I am now actively recruiting for the lab in Michigan. So if you're interested, reach out. Or if you know somebody that might be interested, reach out. Apologies that I went a little bit over time. And I will take any questions if you still have energy.
Wonderful. Thank you so much. So I'm going to manage the chat group.
Yes,
See if there's
if
questions
anyone
there.
has.
But while I do this, please ask questions. Dr. Hood.
That's a great talk. Thank you.
Thank you.
So those knockouts, D1 knockouts have ragged red fibers.
Yes.
So there's some mitochondrial DNA defect possibly there, or
So we
what you
check.
have to do now is take your PGC1
Yes.
overexpressing mitochondria, cross them with that,
Yeah.
and you got rescue.
Yeah, so we checked like the mitochondrial DNA, and we actually checked all on mitochondrial encoded proteins. And what we see is an upregulation of polar MT, as if that mitochondrial DNA is trying to make more mitochondria because there's a dysfunction in mitochondria. But we don't think so far that it's a fundamental problem there. We see it more as a secondary effect of the desmin and everything. But I did show you that the prediction of upstream regulators for those genes being down are transcription factors that we recognize as being like ERR, YY1, all of them connected to PGC indeed. So the prediction is that you could recover the mitochondrial part, but you would not recover the sarcomere part because PGC, as far as we know, cannot do that.
Other questions?
I just wanted to ask about, you mentioned during the tryptophan pathway, you mentioned the quinolinic acid as an MDA agonist and how that can cause certain neurological disorders. What do you think the potential is now that we're realizing drugs like Spravato and esketamine, which are an MDA antagonists, play a role in neuroplasticity and depression?
Yeah,
What is your opinion on that?
Yes,
Because
so-
Canada's been dancing around that for a few years.
really?
Yeah.
So yeah, so that's, I didn't complete the whole story. So in terms of NMD receptor, quinolinic acid is an agonist and kynurenic acid is an antagonist. So in the brain, what is thought is that the play between the levels of these two guys defines excitotoxicity. So there's a lab at KI that has repeatedly reported and seen that patients with schizophrenia tend to have really high levels of kynurenic acid. So that's kynurenic acid, which is seen as neuroprotective. Their message is that if it's too much, then you have dysfunction related. These are mostly ex vivo postmortem kind of studies, but they have this kind of observation. So it's a question of, can we find the right balance to, hit that level of inhibition or excitation between these two things. We've never looked, I know that they are very interested in these ideas of like ketamine that you just mentioned that we were discussing a while ago. I forget about an example of a drug that was developed purely in academia in the lab of John Crystal at Yale, I think, that we had as a guest speaker years ago, and he said something that I never thought about, that they realized that when they were testing ketamine as an antidepressant, that you need the dissociation stage to have a therapeutic effect. So the ketamine is an hallucinogenic drug as well. So if you don't get that dissociation stuff, you don't get the therapeutic effect. So he was saying that most likely if this had been developed by a non-academic environment, you would have stopped there because you wouldn't allow yourself to have that as a side effect, but they really went forward with that. And it's FDA approved as far as I know. So yeah.
It's where-
Yeah, I think depression is such a horrible, debilitating disease that whatever we can get to work, I think we should pursue. That's my personal opinion.
Yeah, Anthony.
Yeah, so you said in the primary myoblasts, you get a phenotype after differentiation or?
No,
during
so
differentiation?
we knocked down HECD1 before differentiation, so as myoblasts, and then we induced differentiation. And instead of making myotubes, if HECD1 is down, they just fuse together. I don't have any pictures, but it's amazing. You get these syncytial enormous cells that are just a agglomerate of myoblasts, which is a very interesting observation. We immediately thought about Eric Olson and Doug Millet's work about the myomaker, I forget the name of all the fusion. So we checked, and at least at first look, it's not a dysregulation of those pathways, but whatever it is, in the absence of HECD1 in myoblasts, they fuse more.
They fuse.
They fuse
I'm
more.
just wondering if there's like a precocious differentiation. There might be a phenotype in the satellites,
There could be. I mean, the other thing is in the knockout mouse, we don't see any phenotype in vivo. We don't see any phenotype that would be reminiscing of more fibers or so. Yeah, but it could be. It could be. I forget if we checked
specific for the knockout, you show specific
for the fiber. Yes, yes.
We have just one question. Sorry, Shiv, we'll come right to you. Ola online, feel free to ask your question.
Yeah, so thank you so much for the wonderful presentation. Like with regards to the screening that led to the identification of XD1, I'm sure you're familiar with some of the screenings that some other people have done, looking for genes that are upregulated in atrophic states. So for example, MAFBOX, MOF1, and some of the other ones. What do you see is the relationship between XD1 and some of these other ligases in terms of the pattern, in terms of the substrates, in terms of the atrophic conditions in which XD1 might be upregulated?
Yes, so very good question. And as I was writing the discussion for our paper, I really thought really hard about that, because the implication, I hadn't really thought about this before, but you don't, in muscle atrophy, you don't tend to have protein aggregation. So what it means is that somehow as you are disassembling the sarcomere, you do it in a way that you can remove proteins from the sarcomere and degrade them. Now on the myopathy side, we know that things that don't polymerize aggregate. So what this means to me is that in atrophy, and I could find some papers in the heart, but fewer papers in skeletal muscle, you have to have sarcomere assembly in order to then degrade it. So it's basically a balance between the assembly and degradation, but in atrophy, degradation wins, basically. So in our screening, we also see that the MRFs, the atrogenes, the MAFBOX, the MUSA1, all of those ligases are increased in gene expression, as many people have shown before. So our we haven't really got into that mechanism, so it's something that I'm really interested in. But our interpretation is that you have an accelerated sarcomere assembly, but the degradation is more prevalent, so it wins. That's the interpretation that we see. So there's some indications that I think, I forget, it's MRF1, there was some indications that it could also target KLHL 4041. So we have not tested if one is better than the other, but whereas MRF1 and atrogene tend to target proteins like titin and things like that, we don't see HECD1 targeting those proteins directly. It's more the nebulin and the desmin and the KLHLs, and then the rest is more of an aggregation or collapse phenotype, if that answered your question.
Yeah, it does. Thank you so much.
Okay, good. Chip?
Great presentation, Dr. West.
Thank you.
I may have misunderstood this point, but the Desmond part of your story, you said that histology was in skeletal muscles.
Yes.
So have you guys looked at the heart histology?
No.
or Desmond's own? No.
No, but that's the project that as soon as we get the mice in, we're going to start that, because everything that I mentioned, these components, so nebulin in the heart is called nebulette because it's a shorter form, but it's the same. Desmin in the heart, there are desminopathies, which are cardiomyopathies. And so we are definitely going to, especially now I'm at the Institute of Heart and Brain Health, so it would be criminal not to test this. As I was telling to some people, my lab never goes into heart projects so much because I really think that should be done by people that know what they're doing. But now I am in an environment that actually has neighbors directly where I am that could collaborate for that. So the next thing is going to be a knockout of HECD1 in the heart to see if there's an effect there. And the other step I think is really interesting is that I mentioned this kind of correlation between HECD1 mutations and neurological disease. And of course, we can't look for the full loss of function because those embryos will never be born. But my question now is, are there any mutations, either in the coding sequence that don't completely abolish the activity, or in regulatory regions for the gene that could explain situations of patients that do have cardiomyopathy, skeletal muscle myopathy, and neurological diseases? So I think it would be an amazing, well, sad, but amazing example of like common mechanisms of disease, and also a very powerful tool for genetics in terms of screening, preterm and whatnot. So we are definitely going to go there next.
Yeah, I was just saying, like, it was an interesting point just because there's something known as arrhythmogenic right ventricular cardiomyopathy, and that's characterized by these desmosomal protein defects. So
Yeah.
it might be something to consider. It's also myopathy.
Yeah, so I didn't know about that particular, but I think David mentioned it. And so, yes, definitely something that we would be looking at.
Very nice.
Thank you.
I was wondering, when you showed the data for glucose tolerance, it looks like they get worse, the effects. Knockout.
Yep.
So is this because of clearance of glucose in muscle, or does it get worse as time goes by? Because you showed data from, I think, 3 months?
3 months, 3 to 4 months. Yes.
How does that, did you have a chance to look into this to see anything, measure anything related to muscle, the oxidative capacity of the muscle, the glycogen synthesis? Does those things change to associate with this alteration in glucose?
Yeah, so it does get worse in terms of glucose tolerance, but we haven't clamped the mice or anything like that. So the protocol that we use for glucose tolerance is with a short fasting of 5 hours to try to not have so much of a liver contribution and ketosis. So we usually think of skeletal muscle as the main and maybe fat a little bit, but we have not done the necessary homework to find out, is it really an insulin resistance, or is it like an insulin secretion or some kind of indirect effect? Since it's a knockout in muscle, we think it's a direct, but it could very well be a secretion of a cytokine that then affects something else for sure. We haven't gone there, but it does also get worse with age. Most things get worse with age, right? I don't know. Try not to get older if you can.
Any sign of calcium stress in the model?
I mean, from the gene expression data, we have spotted that. We have also had our neighbor Johanna Lanner do some ex vivo contraction, and we also see some indications, but again, it's very phenotypic and molecular analysis. We haven't gone down to figuring out what components are. We do see kind of this, like almost a preferential shift of muscle fibers to the oxidative side. So you see a signature that is even in the calcium handling and the bioenergetics, a push towards the oxidative side. But we think that for some reason it's because there's a preferential effect of HECD1 in the oxidative fibers as opposed to the glycolytic fibers. So it could just be very correlated to that.
Yeah, well,
You said calcium, so Arthur was like, "Yep, that's me."
yeah, no, we, sometimes with Julian Ochala, it's
Mhm.
like an myopathy mice, and then when we looked at the calcium, there's no problem.
Okay,
It
okay.
was really a myofibrillary problem.
Yeah. So this one, because it is a combined desmin nemaline problem with the mitochondria, maybe there will be, but we haven't gone into the actual measurements to see if that. There's a collaboration just born right here, so.
Well, thank you very much for giving this
Very good.
amazing presentation.
Thank you for staying until 4:30 on a Friday, so now.
All I saw were
you can-
open eyes.
You can have fun now.
Enjoy the snow, okay?
I know.
And thank you, everyone. And Adam, thank you for all you've done here. Much appreciated. And everyone online, thank you as well. Have a good weekend. There you go. Some compliments here.
Oh.
There you go.